Okay, so in this screencast, I'm going to tell you a little bit about cloning. And the ability to create a clone, this is something that used to be science fiction. But a man named Dr. Ian Wilmot, in about 1997, he and his group changed this idea that cloning is a science fiction idea, and actually created the clone of Dolly the sheep. But since then, the debate on applying this technique that they use to clone human beings, this has been something that's been ongoing. So let's take a step back now and talk about what are clones. Clones are organisms that have identical genetic material. So the DNA sequence, those nitrogenous bases, are going to be in the same order. But even before Dolly, we've seen clones in nature and also in the lab. So identical twins or even identical triplets, all of those individuals are clones of one another. We also find twins we also find clones when we do something where we cut plants and produce clones of each other. So you just take a plant and sort of break it in half. Their genetic material will be identical as well. Let's talk a little bit about history here. So before Dolly was cloned in 1997, scientists thought that you could only clone an entire animal if you used something known as embryonic cells. And embryonic cells are cells that are present in the early stages of an organism's development. We also call these undifferentiated cells. So back in the 1950s, scientists, they were able to generate entire frogs using embryonic frog cells. And cloning a frog is done in a way that looks similar to what's going on in this image. Don't worry too much about exactly what's happening, but again, they're creating a new frog from using an embryonic cell. The reason why they use embryonic cells is because after a small number of cell divisions, then embryonic cells start to change into the different types of cells that an organism will need, whether that's muscle cells, liver cells, blood cells, etc. And embryonic cells, they have undergone differentiation. That's something that, of course, happens after you take those undifferentiated cells. Eventually, they will become differentiated into those muscle, liver, blood cells. Even though each of the cells has the same genetic material, each cell is only able to access the genes that it needs, though, to perform its particular function. So, for example, blood cells will only be able to access the genes that it needs to function. So the egg cell, even though this one is fertilized, will differentiate into all of these different types of cells, and then from it an embryo can form. Before the Dolly experiment happened, this was at a place called the Roslin Institute, they thought that once a cell differentiates, that it couldn't be used to generate an entire organism. Meaning they thought skin cells in a sheep could only generate other skin cells, and not create udder cells and other types of cells that are needed to make up the entire sheep. So at this Roslin Institute, what they did was they took udder cells from a sheep and they basically starved the cells, meaning they didn't give the cells the nutrients that it needed. So this puts the cells into a state that is similar to embryonic cells. And we call this the G0 state. Then they took an egg cell from another sheep, and what they did was they took the nucleus out, which we know contains the DNA. And they do this using a really fine needle, right? So again, this process is called enucleation, and it leaves the cell enucleated, meaning it doesn't have a nucleus. So here's an image showing you that here is our egg cell, here's the needle that we're sticking into it and removing this area here, which is the nucleus. this portion over here is just holding the cell in place so that we can stick the needle in to remove that nucleus. So then, after we've done that process of enucleation, and after we've taken those udder cells and starved them and put them in the G0 state, what we're going to do is try to fuse those two cells. Again, if you're confused right now, I'm going to show you an overall diagram at the end that will show all of this happening, so don't worry too much, we'll review at the end. So again, next, they're using this process to fuse the starved udder cell with the one enucleated egg cell, and they do this using electric shock. And what they did was they made 277 of these fused eggs, and keep in mind that the egg donor, it had a black face, that sheep. And then the nucleus donor, which we're getting from the udder, that sheep had a white face. Okay? So we inserted the fused egg cells into several different black face sheep, which are going to be the surrogate mothers. Right, so there's our enucleated egg cell. We know that that one has that sheep had a black face. And then we're inserting in the genetic material, right, the nucleus, from the other sheep that had a white face, its udder cells. And then we're going to insert that cell into some surrogate mothers. So not the best drawing, but here's our surrogate mother, and now we've inserted this fused cell into the surrogate mother, hence the little bullseye-like shape. That's supposed to be the inserted cell into the surrogate. And out of the entire 277 fused cells that they had, only one of those cells actually developed, and this was Dolly. We know Dolly is that cloned sheep, and she was born on July 5th in 1996. And keep in mind that even though the donor had a black face and the egg cell that was enucleated, that sheep originally had a black face as well, Dolly was born with a white face, which was just like the nucleus donor. So Dolly had the same DNA as those udder cells that she came from. So she was the clone of those udder cells. Dolly surprisingly actually gave birth to a lamb named Bonnie. She actually produced that baby naturally, and also had twins and triplets there. But Dolly, interestingly, developed a whole bunch of diseases. She had lung disease and arthritis, and she died at the age of 6. Some people think that this happened because she was cloned from a sheep that was 6 years old. So maybe she was born with a genetic age of 6 and then lived for only 6 more years, making her live a total of 12 years. So again, still not really sure there. But again, they thought because she was born from a sheep that was not really, really young, that she was born and was already 6, and her cells were only able to exist for 6 more years. So here's a good chart just to summarize what we just talked about. We have the udder cells that we take from a sheep that has a white face. We take that DNA, and then we're going to combine that with an enucleated egg that we get from a sheep that has a black face, right? We remove its genetic material so that it's empty, insert the genetic material from the udder cells into this empty enucleated egg. Those are fused together. We culture them out. We implant that DNA into this black-faced surrogate mother, and then from there we get a clone, which was a white-faced sheep. That is Dolly. And the reason that she has a white face is because the DNA inside of her came from the udder cells of a white-faced sheep. So the second lab that was able to successfully clone an animal came from an adult cell, and that was by a lab in Hawaii. And what they did was they were able to clone mice using something called cumulus cells. We find those in the ovaries. So this was a slightly different process because they didn't actually grow the cells in a culture, but instead they actually used the cells immediately, unlike what they did with Dolly. Also, they didn't fuse the cells here. So basically what they did was they removed the nucleus from the cumulus cell of a brown mouse. So there's the genetic material, the nucleus of that cell. And then they directly injected that genetic material from the brown mouse into the egg cell of a black mouse, hence the black circle around. And so what they did was they tracked the heritage of the mice, and they did that by using coat color. So again, they put that egg cell into a white host mother. So there it is, inserted in. Then this mother, she gave birth to a brown mouse. There's her brown mouse baby. And remember, the brown, that comes from the same color as the mouse that donated the actual nucleus. And so this baby mouse that was born, they named this Cumulina. C-U-M-U-L-I-N-A. And that's named from the type of cell that they use, the cumulus cells. So again, this organism here, Cumulima, was the second second organism that was cloned using adult cells, even though it was a slightly different process than the cloning of Dolly. They also have made clones from Cumulina, and it's interesting because it seems like all of these clones are normal and don't have genetic issues. So, pretty interesting stuff. We now have been able to clone pigs and cows as well, and scientists are hoping that they will be able to use these cloned pigs to actually grow organs that we can transplant into humans. Okay, that's all I have for you now. Bye-bye.